My name's Derek Southwell, and I'm a neurosurgeon at Duke Health. I act as the director of Duke's Epilepsy Center, and I also run a basic neuroscience research laboratory that's studying epilepsy and its treatment. Epilepsy has a number of causes, including brain injuries, infections, and genetic conditions. However, in around half of cases, we're unable to identify a clear cause for patient's seizures. Epilepsy is defined as drug-resistant when a patient has failed to control their seizures with trials of 2 or more anti-seizure medications. Treatment guidelines state that patients who have failed to control seizures with 2 or more medication trials should be referred to a comprehensive epilepsy center, where we offer advanced diagnosis and treatments. Traditionally, epilepsy surgery has involved removal of the area of the brain that causes a patient's seizures. More recently, we've been using laser ablation techniques to thermally damage those same areas. When those types of treatments aren't a possibility for a patient, we've been using newer forms of neurostimulation with implantable devices. Neurosurgeons are often forced to strike a balance between controlling a patient's seizures and preserving their neurological function. Treatments such as resection or laser ablation can, for example, depending on the brain area treated, have impacts on someone's memory or their vision function. In the case of implantable neurostimulation devices, these procedures require us to perform second operations to change out device batteries. They also require complex programming on the part of neurologists and other care providers. Duke is participating in a first-in-human trial of interneuron transplantation for epilepsy. There are around 20 sites across the country that are participating in this study. Interneurons are a cell population that are lost or dysfunctional in different forms of epilepsy. It's thought that the transplantation of these cells can restore or compensate for some of the circuit imbalances that give rise to a patient's seizures. Interneuron transplantation is an entirely new approach to treating epilepsy. It differs from traditional approaches in that we're trying to restore neural circuit function with the transplantation of interneurons, rather than removing or ablating a part of the brain altogether. We transplant interneurons into the brain by using a small catheter that is about the width of a dull pencil. We transplant millions of cells into the recipient brain, which is actually just a very small fraction of the total number of cells that are present in the recipient. The cells are very small, and the total volume of the injection is just about half the size of a grain of rice. This trial involves cell delivery through a single surgical procedure. Duke's study team includes neurologists, neurosurgeons, radiologists, immunologists, clinical nurses, and neuropsychologists. This clinical trial is based on around 25 years of basic neuroscience research. Around the year 2000, it was discovered that interneurons have a unique capacity to migrate and survive in the recipient brain following transplantation. During my PhD studies, I was involved in collaborative work that showed that interneurons can functionally incorporate and modify neural circuits in the recipient brain. We also showed that interneuron transplantation was effective for improving seizure phenotypes in mouse models of epilepsy. Since then, there's been significant interest in producing interneurons from human stem cell populations for use in patients. This has recently led to FDA approval of a human clinical-grade cell product, which is what we're now testing for transplantation in epilepsy patients. We're studying the transplantation of human interneurons into an area of the brain called the hippocampus. The hippocampus sits in the middle part of the temporal lobe, which is a frequent origin of patient seizures. We're asking whether the transplantation of interneurons is safe and whether it's effective for reducing and improving a patient's seizures. We're also asking whether the transplantation of interneurons can preserve or even improve functions such as memory, which are often impacted by a patient's epilepsy. As of the beginning of 2024, 5 patients have been treated as part of the study. Our results are still early, but thus far, transplantation has proven safe in the treated patients. We've also seen encouraging improvements in the patient's seizures. Our next steps are to continue to observe how the initially treated group of patients respond to the therapy. Additionally, we'll be proceeding with the treatment of a larger group of around 30 subjects. Our trial is enrolling patients whose seizures originate from either the left or the right temporal lobe. Patients need to have at least 2 seizure events per month, and they need to also exhibit radiographic evidence of cell loss in the hippocampus. We at Duke are very excited to be participating in this first-in-human study, which is advancing the care of epilepsy. Thank you.